Good afternoon to all of you. First of all, I would like to thank the organizers of the Scientific Services Committee, Professor Rufkar and Dr. Sistla Ramkrishna, for inviting me here to deliver the talk, as well as the session chair and co-chair. So it's very, before I start, this is disheartening to see very few audience, because we are here for students, but that's how when parallel session goes, it's fine. We can continue with that. So today I thought to speak basically the topic what I have planned for students. What are the challenges in extraction, isolation, that is purification of phytoconstituents from the complex mixtures called as extracts. So if you can imagine the extracts that we prepare from plants, or let's say microorganisms, fungi and other natural sources, it's a complex mixture of 200 to 300 compounds. So it's never easy. We work with lot of complex mixture. So that was the idea, how to purify them so that student can have that idea, learn those techniques. And with that, I'm going to discuss one case study on Muraya konegi. All you know, the curry leaf that we use as a common spice in Indian kitchen. So with that, can I have the presentation? Yeah. So the topic what I'm going to discuss, we have published already as a book chapter last year. So before I start with the original topic, just to give you idea, Dr. Amit earlier mentioned the Natural Products and National Cancer Institute, Professor Newman from that particular institute, they published a lot of reviews, and you can say the importance of natural products in drug discovery in the last 6 decades. And if you see the take-home message with this figure is almost 52% of the drugs that we see, small molecule or whole drugs, they are coming from natural products. And out of that, if you see 67% are small molecule, means having molecular weight less than 2000 Daltons. So that is the take-home message from this slide for these scientists who are aspiring to work in natural products. And when we talk about globally, this is the Indian scenario. Dr. Srinivas Reddy is sitting here. He is actually Director CSIR, IICT Hyderabad. So if you see many of these leads, they have come from CSIR lab like CDRI Lucknow. Most of them in that is Central Drug Research Institute Lucknow. They have brought a lot of these you can say lead molecule, and we have worked in this list on boswellic acids in our lab over the last 7-8 years. So now I am coming towards natural products. I have already mentioned what is the importance of natural products in drug discovery and development over the years. So I will not spend much time here. But what is important is that, as I mentioned, the extracts that we have, and we need to, let us say, purify and pinpoint what are the constituents in an extract that are responsible for activity for that extract, which has been evaluated in vitro, in vivo, for different bioactivities. So here it is important, if in order to extract active phytochemicals from plants, it is essential to adopt appropriate extraction procedures. Like appropriate means I see students use simply hot extraction without knowing what you are extracting, because there are many natural products which are present in different natural sources. They are not stable at heat, at higher temperatures, more than 50 degrees centigrade. So if we use hot extraction techniques, they will get degraded. So it is important to know what are the different extraction techniques we should use. And at the same time, we need to think of reducing the reliance on the most conveniently used organic solvents, like we use hexane, chloroform, ethyl acetate. So it's not advisable. So what are the newer extraction methods? That also I'm going to touch here a little bit when I'm talking about what are the challenges in purification and isolation of phytoconstituents. So you see this extraction I have already mentioned. All the B.Pharm students, they are already studying in 3rd year of their B.Pharm, different extraction methods. I will again not go into that. But there are basically 3 types of extraction that generally we use: solid liquid, liquid liquid, and acid-base shakeout that we generally use for alkaloids. So the development of environment-friendly extraction methods for natural resources is currently a popular research topic in the multidisciplinary domains of applied chemistry, biology, and technology. And in that, I will touch this topic later on, that we use deep eutectic solvents or natural deep eutectic solvents. That is a good alternative to that. I have mentioned the commonly used organic solvents. So that I am going to discuss in the last part of my talk. So see, when we work with extract, these are certain interfering components like, fats, waxes, lipids. So we can remove them by several techniques. Basically, we do first extraction of the plant material with non-polar solvents, hexane, chloroform, or petroleum ether, and do the defatting. That is the most conventional approach. Otherwise, we can simply make the initial extract, let's say either with ethanol or methanol, and pass that extract through different stationary phases. That will also remove this, like we can use neutral alumina. That will remove all these fats and lipids from your extract. They will be written on the column, and rest of the compounds you can elute. So this is one of the technique. Or we can use vacuum liquid chromatography. This is another technique which is a short form of open column chromatography, where we can increase the speed of elution by applying vacuum to the end of Buckner funnel, where we do this particular vacuum liquid chromatography and all those things that we use in, let us say, removal of fats and waxes. Then another challenge I will show again in my case study on Muraya konigi. We know whenever we work with leaf drugs, they are rich in chlorophylls, xanthophylls, and related pigments. And chlorophylls, they have wide polarity, starting with solubility in hexane till methanol. So they are not easy to remove from plant extract. So how to remove them? So there are several earlier approaches. People use charcoal, heating with charcoal, but it's non-selective adsorption of chlorophylls, because that will also take out some your compounds of interest. So which is not good. So we need to think beyond charcoal. So there are several other methods. We can use different resins, these anion exchange resins, or we have used also HP20 diane resin in that series, and also Cefadex gels, polyamide, several stationary phases we can use, where these particular pigments are written on the column and your compounds of interest are eluted. That is what I have mentioned on this slide. Then next, another important interfering compounds or group of compounds in plant extract. You take any vegetable drug, leaf drug, or let's say bark drug, they are rich in polyphenols, which are of two types, hydrolyzable and condensed. So these tannins will interfere in many biological assays because they are polyphenolic compounds. They will interact with proteins through the intermolecular hydrogen bonding and give you false positive results in the bioassay. So we need to remove them from our extract. So there are also several methods. The very common method earlier that was used was that we chloroform extract that is prepared from the plant material is washed with 1% aqueous sodium chloride, and the top phase can be discarded, and then the chloroform phase is further dried on anhydrous sodium sulfate for a few hours to remove tannins. Or we can use these different stationary phases, polyamide, collagen, Cefadex LH20 or silica gel. But in this, most commonly we use Cefadex gels. They are very, very expensive. The alternative to that is actually polyamide, which is quite cheaper as compared to Cefadex LH20, where again we can remove the tannins from the plant extracts. And also when we work in the laboratory, avoid using plastic containers, because if you are working with organic solvents, these organic solvents like chloroform, ethyl acetate, will leach the plasticizers from these plastic containers, and dioctyl phthalate is a common plasticizer compound that is leached out from these containers, and through solvents they come into your extracts. So they are interfering compounds. So don't use plastic containers, try to use glass containers in your experiments. So with this, I come to the case study. Now you know that curry leaf, I have shown the picture. Everyone in the hall sitting here is familiar with this, I am sure. Yes or no? So now you are thinking, so what I am going to do with this? So we know that it's in Ayurveda, it is called as Meetha Neem. Kadu Neem is Azade recta indica, neem tree. This is Meetha Neem, that is called as actually Muraya konigi in botanical name, English name, and in Sanskrit, Ayurvedic literature, it is mentioned as mithanim, means you can chew the leaves. So there are many benefits mentioned in Ayurveda, and when we look at the literature, the extracts prepared from leaves are shown to actually decrease the high blood glucose levels in experimental animals, as well as there was anti-hyperlipidemic activity and other so many antioxidant and different other biological activities are reported. So based on that, we thought of exploring this curry leaf, and curry leaf is powerhouse of these carbazole alkaloids. That's why I have mentioned alkaloids in diabesity. You know that diabesity is nothing but the diabetes associated with obesity. See, 60% of the obese people, the literature says, are going to develop type 2 diabetes. So that is the reason we can also see when there is obesity, there is a chance of developing type 2 diabetes. And we see now India and many Western, Eastern Europe, Asian countries now are becoming diabetes or it is going to become capital of diabetes. Now, disease of waste has come to our part, and our change lifestyles, you see, this diabetes is seen now in young people with the age of 40. Like we, doctor advised, do HB1AC and blood glucose level and see where you stand. So this is the real risk, and we have so many herbs. This is just one. We are working with those, but I thought of showing one. So these really help to regulate your lipid profile as well as blood glucose level. So let us see with that. And these are different genes that are associated with adipogenesis, lipogenesis that we are working with as the mechanistic aspect of the extract, fractions and compounds. So these were earlier studies done our lab when we started this work. Preliminary studies with the safety, that is the toxicity, as well as the anti-obesity activity in rat model of obesity. So you can see there was no toxicity with the methanol extract, dichloromethane extract that were prepared from the leaves of curry leaf. And accordingly, further if you see these total cholesterol, triglyceride level, they were decreased after the treatment with these extracts, as well as the bioactive marker in the extract is mahanimbin, which is a carbazole alkaloid. There also you can see there is significant decrease in total cholesterol and total triglyceride level when it was tested in animals. And this is a study with mahanimbin at low dose, that is 2 mg per kg per oral and 4 mg per kg per oral. So you can see here all these lipid parameters, they have been significantly reduced. If you compare with the disease control group, that is high fat diet induced obesity in rats here. So HFD plus high dose and low dose, and you see the control HFD, that is the disease control. So I have compared those values with the high dose, that is 4 mg per kg per oral of this compound mahanimbin. I will show you the structure. So what you see, very important observation, what I want to show, don't know. You see there at the end, the high density lipoprotein levels, that is good cholesterol, that has been increased in presence of the mahanimbin. So this was our first observation. This is exciting because I don't see any drug in the market, which is anti-obesity drug, that increases HDL. So this attracted us to take the work further and try to develop a product from the curry leaf, these particular observations. So before that, as my theme is extraction and isolation. So this was one paper again published from IIM Jammu a few years back. They did this isolation of 11 alkaloids from Muraya kunigi leaf. So we followed that paper and did this extraction. This is a scheme, but what I want to show, like you see, we have not used hot extraction method. They have used methanol and chloroform mixture at room temperature, and that was 7:3, chloroform 7 parts, methanol 3 liters 3 times. At room temperature, extract was obtained, and then it was subjected to column chromatography on silica gel. 5 pooled fractions were obtained, and then in these fractions, Now each fraction had several compounds. Now you see all these fractions were taken together and a series of chromatography methods have to be utilized. Repeated column chromatography, sometimes changing the stationary phase from normal phase to reverse phase, going towards the preparative or semi-preparative HPLC, as well as preparative thin layer chromatography, and the recolumns that we do and try to get the compounds. Now you see in this particular scheme, fraction 1 gave 3 major compounds. Mahanimbin, I just discussed, we had isolated also in 50 gram quantity from 1 kg leaf already in our lab. Then gerinimbin and O-methylmuramine A. So these were the major alkaloids, and there were several minor that you see, coinimbin, conigicin, then muraconin and others. So we followed this methodology, prepared the extract, and first thing in natural product, what is required to be done is run LCMS. So unless and until we run LCMS, we will not know what are the compounds present in the extract without isolation. Just try to identify based on literature. I showed that 11 alkaloids. So we could identify those in our extract by using LCMS. So what LCMS information gives us? It tells us about the molecular ion peak for the individually separated peak in HPLC. So this is total ion chromatogram that we observe in LCMS and try to identify mass wise for every separated peak we get m by z value, that is the molecular ion peak information, and from that we know what is the molecular weight, and we can compare what are those reported compounds with the same molecular weight from the same plant, and we can say, okay, this is the compound. Tentatively we identify, we get the idea. So I wanted to know whether mahanimbin, coinimbin, conigicin, gerinimbin and so on, whether they are present in our extract, and they were present. So table I have shown here. So all these alkaloids were isolated based on their m by z ratio, and accordingly we had isolated these 4 bioactive compounds. But before that we also determined, now another important thing, the content of phytoconstituents in plants, they do vary. They do vary depending upon geographical, seasonal, then genetic and other factors. So here we wanted to see that the seasonal factor, which is the best season. So what we find through our study on our campus, Naipur Mohali Medicinal Plant Garden, we have collected the samples throughout the year, and what we find, the leaf sample collected from Muraya konigi plant, curry leaf plant, during April, May had the highest content of alkaloids, and we see that mahanimbin is present in 0.51%. You see the content I have given. So accordingly we have chosen and selected that the plant material only to be collected during April, May, that will have maximum content of alkaloids. So, and this only not depend on geographical, as I mentioned, or seasonal. It also depends on soil, rainfall, then the amount of sunlight during the day that is required. So those analysis still to be done, but soil also plays very important role in the formation of secondary metabolites in a plant. So those studies are required to be done. And then this is a process now. Now you can see what I have shown here, HPTLC on the right hand side. So I don't know, this is not working here. So if you see, I tell you there, the original extract is ME. Now you see all these red color bands. What are these? These are chlorophylls. So I have mentioned how to remove chlorophyll from the plant extract. Now you see on the left and right hand side of that particular methanol extract, there is no, these red, pink color bands have disappeared. So that means we have successfully removed those chlorophylls and prepared the chlorophyll-free extract that was enriched with the alkaloids of interest. These are carbazole alkaloids that we wanted. So you see AEF1, AF2, 3, 4. 4 trials we have done at 100 gram scale, 100 gram methanolic extract, and we see around 51% yield. From 100 gram we got 51 gram of these enriched fraction that you see devoid of chlorophylls, other interfering compounds, and you see at the top that purple color compound, that is mahanimbin. So that is enriched in the fraction, and we could successfully remove all those interfering compounds, and we have already filed patent last year and on this particular work process, and then the formulation that we are preparing out of this enriched fraction, alkaloid enriched fraction prepared from the methanolic extract of Murraya konegi or curry leaf. And we have already prepared the capsule formulation, taken the trial on pilot scale. We have already checked with 500 capsules batch, and we are getting consistent result with this alkaloid enriched fraction with 1 or 2 excipients for capsule formulation. We could easily make this formulation, and all the, let us say, mechanistic aspect and all that I will show further. So these were the 4 compounds. Apart out of the 10, 11, we identified. Others have also been isolated. I haven't shown here the structure. I am talking about the product development. You know that either we can go for phytopharmaceutical product or a nutraceutical product. So you know that in 2015, Government of India has actually notified a new drug regulation called as phytopharmaceutical regulation, and this is nothing but an effort to promote the product development from single medicinal plant. So it is a very good initiative by Government of India. Like if you see CDSCO website, Standard Control Drugs Organization, under that you will find this regulation. So this is important. So we thought of taking it to either of the route, whichever company takes it, whether phytopharmaceutical or nutraceutical. We are ready with all the data. So these were the 4 bioactive markers that we have isolated from this extract: mahanimbine, girinimbine, coinimbine, and conigesine. And their quantitation also I have mentioned by using different extraction methods. And this is the, like we wanted to do anti-obesity and anti-diabetic activity study. So it is important in vitro you assess the cell viability. Amit also showed earlier that we have to see the cytotoxicity, and we have to ensure whatever observed activity is there, it is not due to cytotoxicity. So for that we are doing cell viability study. So all these alkaloids were, you can say, not cytotoxic to the cells. When we were tested at different concentrations, you will see starting with 10 or 15 micromole to 30 micromole. These were safe, having cell viability more than 80%. And we have used these 3T3L1 adipocytes. Pre-adipocytes, they are converted into mature adipocytes, and in that we study how much lipid accumulation is reduced in presence of our, let us say, test compound, that is the alkaloid enriched fraction and bioactive compounds. So you can see here the best result what we see is with the fraction. Alkaloid enriched fraction 37.4. So if you compare with all other values, it is the lowest. That means it is logical to take it forward as a mixture, not as a pure compound. So with this we started further continue our study, and we chose this particular animal model. This is C57BL6 mice, which are the genetic mice where the data generated you can directly extrapolate to humans. So that was the idea, to take this mouse, and once we feed these mice with high fat diet, they become literally 25 gram mouse. It become a bulgy having 35 to 45 gram weight. They actually got it over the high fat diet, and we have to see how much that is decreased when we give the treatment. And this is 13-week protocol that we have done. 13-week we have fed the mice with this alkaloid enriched fraction. Now you can see here we have used at 2 doses, rather 3, 50, 100 and 200 milligram per kg per oral, and out of that 100, the dose of 100 mg per kg per oral showed the best results. So we have also done the toxicity study as per the OECD guideline 407, that is repeated dose acute toxicity study in rats for 28 days, and what we find, up to 1 gram per kg per oral, this fraction is safe. There is no toxicity. No toxicity signs were observed. And now you can see here another result. I have shown you earlier mahanimbine. Their HDL level was increased. Now you see the HDL level here. From, you consider the disease control, 28, you see it is less, and it has increased to 37, further to 42. So that means we have already a very active extract that increases HDL, which is very good thing. That is what we want when we want to control the lipid profile. We need to see that reduction in LDL, increase in
Please
HDL.
conclude,
So
sir.
this was the major highlight of our research, that we have already filed patent along with process and all that. So we are talking to two, three companies currently. They are interested to take this product further into the market
Sir,
under the area of dyslipidemia.
Can you conclude quickly?
Yeah, just this study only. And this is a mechanistic study we have already done with the different gene expression that you can see for liver and VAT, all those lipid related biomarkers as well as inflammatory markers. They all were down regulated when we have done this study for 13 weeks in HFD mice. Okay, so last part I will not able to complete this deep eutectic solvents, but see, apart from whatever challenges I tell you, there are major phytoconstituents. You can isolate them without chromatography. I want to show you two, three examples. There are several we have done in our laboratories over the years, and you see this is one compound, diaxophenylene derivative, that you see baracol, that is present in this Indian cassoid tree called as Senna siamia. So leaves, you know, it's a Fabaceae family. Many Senna trees have yellow color flowers. If you know Senna, you have studied in your bee farm, isn't it, Senna? So it is same family. That is Leguminosae or another family under that. So here we have taken this dried leaf of Senna siamia, and then it was sonicated with 0.5% sulfuric acid in water for 2 hours and filtered. And then this aqueous acidic phase was made alkaline to pH 9 by saturated solution of sodium carbonate and partitioned with dichloromethane. So dichloromethane layer was concentrated and shaken again with DCM with water and filtered. And after that, in the filtrate, we see this compound was precipitated. So pure compound, it was 95% pure. We checked with HPLC. So without chromatography, we can isolate, number one. Number two, this all you know. This is called as harat, you know, very important ingredient of Triphala. So that contains many complex tannins, hydrolyzable elagitannin. So it is such compound, which is cebulinic acid having molecular weight 956, and there is one more, cebulagic acid 958. These two are together, but this is major. So we can extract it, purify without chromatography. You see that we have taken 800 gram of dried Terminella cebula pericarp, then done this oxalate extraction with acetone, filtered acetone extract that was concentrated under rotary evaporator to 1/7, then finally diluted with water to initial volume. And after this stage, we see precipitation of elagitannins. And that was left undisturbed for 48 hours. We got the fine white colored precipitate that we have separated, and that is nothing but actually cebulinic acid. You can see 18 gram from 800 gram without chromatography. You can isolate. And last example is, this is, you know, a mixture of vitamin P you see in citrus fruits. So what is this plant? Citrus limata, you know that. This is sweet lime, Mosambi as we say. This is the peel of this Mosambi that was taken to isolate this flavonone glycoside called as hesperidine, which is nothing but the, if you see at 7th position, we have a disaccharide called as rutinose. So this is iriodictyol 7-O rutinose, that is called as hesperidine, and this is very important flavonoid having a lot of biological activities including antioxidant, anti-cancer and so on. So that we had isolated in 13.57% yield, 190 gram from, you can see, the 14 kg of fruit peel of citrus limata. So there are many such natural products you can isolate without chromatography. I have shown you very complex procedure for carbazole alkaloids. These are very simplified procedure. So this is what the challenges we face, but I think it is not like that. It is not doable. It is very much doable. You have to do a lot of trial and error, have patience, do not give up. Then definitely you will see that at the end of the day you will be successful. So this is what is required in really at the bench level in natural product research. That is what we expect from the students and scientists to do with that. So this is a scheme. I will not go into detail, but you see, without chromatography, we have tried on small scale first. Then on the large scale, that is the isolation of this glycoside hesperidine. And then actually, I will not go into now, my time is over. This is another important thing that we have to use this deep eutectic solvents, where generally we use hydrogen bond acceptor and donor together with the complexing agent, and there is a hydrogen bonding in between these two components which decreases the melting point, and we have very, you can say, solvent system that can be used for extraction of different types of compounds like phenolics, alkaloids, and terpenoids from plant extract, but lot of optimization need to be done. So this is a current trend in natural product now extraction and isolation that all you need to follow. Just this is the thing what we have, and this is a general formula. When I was talking about hydrogen bond acceptor and donor, we can take and we can have these particular deep eutectic solvents, these are natural deep eutectic solvents for the extraction of plant compounds. So this is what it is actually. Let me see. These were the things. Now you can see on the left hand side, these are the halide chloride salts. On the right hand side, different natural acids. We can use hydrogen bond donors. These are acceptor, and we can make these eutectic mixtures and use for extraction of compounds. And these are two examples. Now you see choline chloride lactic acid was very useful in extracting glycerizic acid from Glycyrrhiza glabra. You know very well this compound, and other the alkaloid example anyway. So what is the advantage? Green chemistry strategies forward in organic synthesis as well, and high tunability. So with this I finish my talk, I think through shortage of time, but lastly I want to show that this one of my students got this award, and here we are at the very most green campus at Naipur Mohali, the greenest area in Tri-City, Chandigarh, Panchkula and Mohali where we are. So this is our natural product field laboratory where all the processing of herbal drugs is done. And this is my research group of PhD students, and this Mrudula Singh has done this work on Muraya Konegi. So with this I thank you all, and there will be if one or two questions I can answer. Thank you.
Nice one.
Thank you.
I just want to draw your attention to one point, sir. Chlorhexidine is used in oral care cavity.
Which one?
Chlorhexidine mouthwash.
Oh, okay.
Okay. But chlorhexidine mouthwash being synthetic causes also sterilization. As a consequence, oral microflora is disturbed.
Right.
So the market gap today is to have, if possible, a herbal oral rinse which
Right.
will not disturb the oral microflora and reduce the pathogens. So
Okay.
in that context, you can see if your curry leaves will have any curry leaf extract.
Because it has
will be used.
essential oil. Yes, definitely. It's going to work. True, true. Yeah.
The last
Yes.
question.
One more. Yes.
Good evening.
Yeah, good afternoon.
In animal studies, what is the reference drugs? The anti-diabetic. We didn't keep the reference drug in that groups.
Which reference drugs?
Uh,
For?
Comparison.
Acha, for anti-obesity activity.
Yes, sir.
We haven't used any, but there are few on the market. That is already lot of products in the market as anti-obesity. So we compared with that as a standard. Yes.
Okay. You compared with the marketed herbal drugs.
Yeah.
Marketed
Little
herbal
louder.
drugs.
Yeah.
Garcinia, Cambogia.
Yes. Yes.
Okay.
That is a reference standard, herbal standard we have used.